Next up, we have oral presentation 3, Fitchburg, Massachusetts, Mission 11 flight experiment.
Hello, everyone. We're the Mission 11 team from Monty Tech in Fitchburg, Massachusetts. I'm Samuel Poulin. That's Felipe Rodriguez, Caleb Van Hillo, and Tanner O'Neill. And so we chose to do our experiment focusing on the bacterium Alcanivorax borcumensis, an oil-degrading bacteria which we hope will be used in the future to help bioremediate oil, possibly in environmental cleanup or waste removal systems. And so our predictions before we sent this into space were that, first of all, it would be successfully activated and deactivated once in space, in orbit, and that it would have increased growth and higher oil consumption.
So there was a lot of pre-preparation that went into the experiment. So we used a Type 3 FME tube and clamp system in order to control the activation and deactivation of the flight sample. Both samples were sterilized and contained 1 ampule of the freeze-dried Alcanivorax borcumensis, a growth inhibitor, and a growth serum. The analyses we performed included an optical density test, an oil filtration, a microscopy analysis, and a mass pellet test. We did serial dilutions on 1 milliliter of each sample 6 times and ran the testing at 600 nanometers. And as you can see in the photo, when completed, the ground sample had a visually higher turbidity than the flight sample. And the results of our testing showed that the ground, both diluted and undiluted, had a greater amount of cells per milliliter compared to the flight samples. And this suggested to us that the ground samples were more effective in terms of cellular reproduction.
So in the image here, you can see is the photo of the microscopy of both the ground and the flight sample. Both samples had a very high density of cells, but the ground sample had what we assumed to be either cell debris or a contamination and also rod-shaped clusters, while the flight sample had, interestingly enough, crystalline structures, which we attributed to either possible cell structure changes or changes in the growth inhibitor. We conducted a mass pellet test after running our samples in a centrifuge and drawing off the supernate. You can see here the values. The ground sample had a much higher mass than the flight sample. And that can be attributed to the fact that it's evidence to suggest that in microgravity, our bacteria reproduces at a slower rate. The next test we conducted was an oil filtration test where we took the supernate and separated it through multiple layers of filter paper. And there was a 3.175 difference between the two masses. So this means that even though the ground sample reproduced faster, the flight sample still metabolized the oil faster.
Benefits from the knowledge we gain from these experiments include a potentially environmentally friendly solution to bioremediation purposes, which include, as in this picture, the deep water horizon oil spill, in which this specific bacteria was actually used to help degrade the oil that was spilled into the ocean. It also, because in comparison to the ground sample, the flight sample had a lower reproduction rate and a higher metabolism, so in microgravity, in space environments, it's possibly more useful rather than on Earth. Future questions we had include why specifically does the flight sample have a higher metabolism but reproduce less, and would this continue if the flight sample were brought back down to Earth? Also, would microgravity affect the bacteria's ability to produce enzymes such as ALK-B1 and ALK-B2, which are responsible for bioremediation.
I'd like to thank our mentors, Dr. DeAngelis and doctoral candidate Gina Chaput. And I'd also like our sponsors, Mass Science Grant, Money Tech, and Subaru and Casis for giving us this opportunity with their help with financing. And so now we have an opportunity for questions.
I'm John. I'm from Stamford, Connecticut. And just what do you hypothesize is the reason for the better metabolizing in microgravity? And then how can we translate that to make the process more efficient on Earth?
So it's really hard to tell at this point with so many different factors about what could actually cause the difference in metabolism rates. It's as well to remember that we did have a possible contamination or a rapid cell growth within our ground sample. But if it does metabolize faster for whatever reason, that could be and if that effect remains when the sample is transported back to Earth, that could be used to create almost an enhanced version to help with the bioremediation and to make the process more efficient.
Thank you.
I'm Gabriel from Burleson, Texas, and you said on your ground experiment you may have had it contaminated. Did you ever try redoing the experiment with the data you, with your timeline you had on the ground?
Well, in terms of the contamination, we were uncertain as to, it is a possible contamination. We're uncertain as to its origin. As we stated, everything was sterilized and we took the proper procedures to make sure contamination did not occur. We did not have the opportunity to redo the experiment, however, but yeah, we would definitely and love to in the future.
Okay, thank you.
Thank you.
Oh, come on up.
Hi, guys.
What's your name and range?
All right. My name is Michael Gray, and I'm a computer scientist from University of Texas at Permian Basin. My question is, could you clarify a little bit what you mean by oil? Would that be is that a crude product or?
Yeah, it's a crude oil specifically, specifically ones that contain N-alkanes, because that's what the Alcanovorax porcumensis bacteria actually degrades, is the alkanes.
Okay.
Thank you.
Hi, I'm Jenna, and I'm from Wisconsin. I'm an English teacher, so forgive me if this question was already answered, but can you foresee any medical implications of bacteria growing more slowly in space?
Could you specify whether you mean the application for this bacteria specifically or just in general?
Well, I don't know. Like I said, I don't know about this in general. Let's say in general.
So in general, the trend is most bacteria reproduce faster in space because of the less stress on the bacteria themselves, which is what makes this case so interesting especially. But to apply to the medical field, there are plenty of helpful bacteria that if grown in accelerated processes in space could be reproduced at a faster rate and brought back down to help with whatever that is.
Yeah, that makes a lot of sense.
Thank you, guys.